This section explains what synthetic biology is and why it matters at the level of a single structural idea. Start with engineering itself. Engineering is the move that makes a substrate composable. You hide each part's complexity behind an abstraction with a measured interface so that parts combine predictably. That is the whole trick. A known interface lets you snap pieces together without holding every internal detail in your head. Now, the thesis. Biology is already the supreme manufacturer. The bacterial lawns of Menlo Park outproduce Intel's global chip output by a factor of 16-to-1 by mass, and the pinecone and the abalone build with an atomic precision we cannot yet match. Synthetic biology is simply that same composability, move applied to living match. Concretely, that means you read, write, and standardize DNA as though it were a programming language. You black box the biochemistry behind standardized parts, the bio bricks, so a designer can use a part without re-deriving its chemistry. Then you run the design, build, test cycle over those parts. The framing here is upskilling biotech, raising what a practitioner can do rather than stripping skill away. The tools add reach, they do not diskill the field. One boundary reveals the whole structure. Life mutates. Because it mutates, the composed part drifts away from its specification over time, and that drift is the tension the rest of the discipline has to manage. This section explains the single move behind every revolution in making. Every such revolution is the same move. A class of matter gets reclassified as programmable stock. Stone first, then smelted metal, then doped silicon. Elphick and Endy's opening wager is that living matter is next. The primitive is substrate redesignation, and the chapter's whole job is to install it for biology. The anchor does the persuading before any argument. Menlo Park's lawns out manufacture Intel by mass. The world's silicon chips are 6% of what the town's gardens build each year. Atom-precise, self-assembling, self-reproducing, solar-powered, and biodegradable. A pine cone is a seed dispersion subcomponent. An abalone lays calcium tile by tile. If that machinery can be engineered rather than merely admired, life joins the palette of materials. This section explains how art counters the engineering mindset in synthetic biology. The single engineering paradigm is a lossy compression of the living. Life becomes raw material, its irreducible messiness discarded so that it can be typed in, standardized, predicted. Katz and Zur answer this, not by walking away. Refusal would mean longing for a romantic past where humans and nature lived in harmony, and that past is both unattainable and nature lived in harmony. Their counter is occupation. They enter the same lab, wield the same biobricks, and use those very tools to re-inject the ambiguity the compression threw away. The mindset signature is the either-or, a certainty, seeking intolerance of uncertainty. Art's countersignature is the deliberately messy artifact, an object that is neither utopic nor dystopic. Arnold's admission that there is no such thing as a standard component seals the point. The compression is never lossless because life resists it. And so the chapter's telos comes into focus, keep engineering from monopolizing life. This section explains one paradigm now spreading and the temperament that marks it. Life gets reframed as raw biomatter shaped by engineering logic. Its signature is a temperament rather than a technique, an intolerance of uncertainty, the either or reflex that wants control and calls ambiguity error. Katz and Zur name this the single engineering paradigm. They counter it by entering the same laboratory and turning its own tools to works that stay messy and contestable, neither utopian nor dystopian. Rather than retreating into some untouched romantic nature, a reaction they hold empty. The answer to a mindset that would monopolize life is the deliberate keeping open of what engineering closes, not a rival mindset. Refusal concedes the space. Occupation contests it. This section explains one primitive. A borrowed word can reveal what a discipline forgot to import along with it. Two disciplines can share a word and mean opposite things, and the gap between those meanings is diagnostic, a signal rather than noise. Ginsberg's lever is the word design, a false cognate. Synthetic biology borrows the term but imports only engineering's sense of it. In that sense, design means necessity, the bridge that must hold, the defined problem solved more cheaply and disskilled. Her own sense is possibility, life as it could be, projecting new futures out of existing matter. The chapter's whole move is to wield the second meaning against the first. Design becomes a critical lens that asks what a living technology could be for, rather than the service that translates a technology into consumable stuff. The primitive is this. A discipline's imported vocabulary reveals what it forgot to imported vocabulary, reveals what it forgot to import with it. This section explains how a single word, design, is doing two incompatible jobs at once, and how that equivocation becomes the whole lever. In synthetic biology, design means engineering design, solving a defined problem, the bridge that must hold. Design, as Ginsberg practices it, means possibility, life as it could be, framing futures that have not yet happened. Synthetic biology claims the second meaning and imports only the first. So it promises to disrupt nothing, bacteria brewing the same fuels and the same plastics in vats instead of wells. The chapter's generative move is to run that equivocation as a critical lens. A borrowed word smuggles in the telos of its home discipline. Naming the substitution is what exposes the disruption as merely nominal. This section explains how nature earns the word design. Design moves from cause to signature. It becomes the property of a system pressed against a physical limit, the mark of optimization rather than the trace of a maker. ND sets two readings against each other. The first is the alien authorship possibility, the idea that the nested genes of the Phi X174 virus carry a message from an author outside nature. That reading is flatly refuted. The second, accepted reading is that evolution spontaneously generates systems optimized to their constraints, so that apparent design needs no designer. Two anchors carry the point. The fly's eye sees at the limit of photon shot noise. The charged backbone of DNA tolerates base swaps without losing function. Each one is design read as optimization to abound. This chapter is the book's affirmative pole. Its generative move is this. Once you define design as constrained optimization rather than intention, nature qualifies, and the engineer's ambition becomes competing against billions of years of it. This part explains the first structural primitive. Selection, pushed to a physical wall, looks exactly like design. That is Indy's whole move. The fly's eye computes at the photon shot noise limit. DNA's charged backbone lets any base swap for another with chemical impunity. The genetic code itself is tuned so that random mutations tend to land on survivable amino acids. This is optimization so extreme you could not engineer anything better. Design here means the signature. A selection process leaves once it saturates the possible. No author is involved. The primitive is this: wherever a natural system sits at its physical performance ceiling, designed becomes the accurate word with no designer required. The chapter is the affirmative pole of a staged dialectic. Chapter 5 answers the claim that there is no design in nature. This chapter is the ground against which synthetic biology's own designs get measured. This section explains why nature contains no design. Design is not a property of an object. It is a predicate of the agent behind it. Design factors through values, through intention held by someone who cares. Shifter's move is to locate design in the maker's volition, not in the artifact's form. Nature's exclusion is then immediate. Evolution is a series of occurrences with no agent to bear awareness. So saying nature designs is personification, convenient but false. This chapter is the book's pivot. Once design attaches only to value-driven agents, synthetic biology's phrase, designing with nature, stops meaning adding novelty and starts meaning importing values into a domain that had none. You can predict the rest. Every later chapter inherits the question, the reframing forces, whose values? This section explains what design actually is, and why calling nature a designer smuggles in a choice nobody has examined. Design is a predicate that presupposes an agent, a claim about someone acting, rather than a property that belongs to things, and the mark that truly distinguishes design is values. Intention is the usual candidate for that mark, but it is not the one that holds. Shifter strips away every candidate mark of design from evolution. Evolution is blind, it surveys no alternatives, it has no planner. What remains is the one mark that cannot be explained away as metaphor. A designer applies norms, mores, principles. So the phrase nature designs is convenient shorthand, never fact. The mangrove and the pine cone do their work without a blueprint. The chapter's job in the book is a conversion. Synthetic biology makes a boast, design with nature. That boast becomes an admission. Synthetic biology injects values into a domain that had none, and those values are presently borrowed, unexamined, from engineering. The polemical title then collapses into the only question left standing: whose values and why those? This section names the primitive behind design evolution. Design an object that lives, and it stops being an object. It reproduces, mutates, dies, escapes into ecosystems. The artifact keeps evolving after you set down the plan. That collapse is the primitive. Take Ginsberg's growth assembly, a herbicide sprayer grown as 7 plant parts. It carries its surprise in its own flaw. Forcing manufacture onto life still designs out the very properties, self-defense and adaptation that evolution supplies for free. So design must itself evolve. It moves from design of biology, which imposes engineering logic and edits life away, to design with biology, where life, death, reproduction, and evolution become materials rather than defects. This chapter opens the book's turn from asking whether there is design in nature to asking what makes biological design good. This part explains the pivot at the heart of the book, and it turns on a single preposition. Synthetic biology as it is practiced, designs of biology, it treats life as raw material onto which an external engineering logic is stamped, and it treats biology's own properties, death, reproduction, evolution, and self-adaptation as noise to be designed out. Ginsberg's move is to design with biology, to make those very properties the medium rather than the enemy. Her growth assembly, a herbicide sprayer grown as 7 plant parts, carries the argument by failing at it. It lets human design subsume nature's own, doing clumsily what evolution streamlines. This is the thesis seeding the book's arc. There is good design, but no perfect design. And good for biology means keeping the system's generative logic doing the work rather than engineering it away. This part introduces the first structural primitive of Chapter 7, biologic. Stop copying nature's shapes. Run its programs. A xylem cell carries no stored structure. It executes a morphogenetic algorithm that renders support in response to whatever envelope constrains it. Benjamin and Federici seize that program and feed it human-drawn boundaries, letting the cell solve shapes no designer specified. Bacteria give the mirror case, pattern formation plus material deposition as decentralized manufacture. The primitive of Chapter 7, replicate the logic of nature and not its form, reframes design from choosing fixed forms to steering a generative process under shifting, partly unknowable forces. Imitation samples, only the forms, evolution already fixed. Harnessing the program opens the whole design space. Those constraints never let nature visit. This section explains the single primitive the 7-body chapter turns on. Biomimicry copies nature's frozen output, the sponge's fiber, the earthworm's gait. Benjamin and Federici make a move 1 level up. Do not copy the form. Run the program that generates it. A xylem cell is a decentralized shape solver, a set of local molecular rules that render structural support to fit whatever boundary the cell inhabits. Extract that morphogenetic program, feed it a human-designed envelope, and it solves for shapes nature never met. The chapter's compression, in their own words, is to replicate the logic of nature rather than its form. The primitive is the lift from artifact to generator, a photograph versus the camera, an output versus the algorithm that produces it. Once you hold it, every biodesign case predicts itself. This section explains how metaphor quietly carries a logic from one material to another. A metaphor is a silent scale, crossing. Call DNA a circuit, and you have already asserted, without testing it, that the logic valid for silicon at laptop scale holds unchanged at the scale of nucleic acids. That is the chapter's whole move. Scale is the act of translating a logic across materials, not mere size, and metaphor is the gateway that smuggles that translation past inspection. Benjamin and Federici's xylem work is the anchor, architecture's optimizer meeting biology's growth. The surprise that shifts your model is that language has material consequence. Think cells as circuits and you will build on biology rather than with it, until each metaphor becomes a question rather than a given. Engineering's logic is imposed rather than earned. This section explains why a metaphor is never innocent, and what that costs a field that borrows one. A metaphor is never decoration. It smuggles in the logic of its home domain, and that logic is scale-bound. It is earned where it was forged, and it becomes unwarranted the moment it crosses scales. That single move is the whole chapter in shifter's scale and metaphor. Calling DNA a circuit imports silicon's logic into the cell, as if size metaphors silicon's logic into the cell, as if size, material, and behavior were free to translate. Biomimicry copies a leaf's form into a building while leaving its why behind. Synthetic biology copies the form of electronics into biology the same way, taking the shape and dropping the logic that gave the shape its meaning. The generative test asks one thing: does the source logic survive the change of scale, or does it merely look like it does? Until metaphors become questions, the field keeps designing on biology instead of with it. This section explains the chapter's real argument, that a designer and scientist collaboration meant to imagine living products discovers its actual prize somewhere else entirely. The collaboration sets out to picture living products, microbes grown into a cup, skincare secreted by tailored flora. Its true find lies elsewhere. The chapter's thesis is an isomorphism, the funnel that IDEO drew to tame design's divergent, then convergent cycles describes unchanged. The scientist's hidden practice, then convergent cycles describes unchanged. The scientist's hidden practice. That hidden practice is what Jacob called night science, the untidy creative struggle that the official day science account erases. Design's gift to synthetic biology is not the concepts. It is a map that renders that struggle explicit, teachable, demystified. So, the chapter's place in the book comes clear. The speculative products are the pretext. The payload is that two disciplines apparently worlds apart run one shared creative shape, and design happens to have already charted it. This section explains a hidden shape shared by science and design. Strip the polish from a scientific paper and from a finished product, and the same contorted funnel surfaces underneath. A funnel of expansion and contraction, ideas bred in bulk and then culled by testing. François Jacob named the two halves. Day science is the linear published order, pretending the conclusion was known from the start. Night science is the actual jumble of doubt, sweat, and inspiration that generated it. The chapter's move is to notice that IDEO's diagram design process and the LIM Lab's night science are the same generative machine. Only one discipline has bothered to draw it. Science and design converge in the messy process that both finished artifacts conspire to erase. This section explains why the very act of turning a craft into an engineering discipline is what drains the creativity out of it. The same move that makes something an engineering discipline, abstraction, standardization, division of labor, is the move that strips its creativity away. Elphick reads his own profession as the cautionary instance. Engineering was once imaginative, tinkering, and it industrialized into silos of designer, engineer, and technician, silos of designer, engineer, and technician, subcontracting inventor of other people's inspiration. Synthetic biology now applies that same formalization to living matter, and it stands to inherit the same impoverishment. Here is the turn that shifts your model. The constraint that makes a discipline powerful and scalable is the very same constraint that forecloses its unanticipated futures. Preserving creativity is what keeps the space of possible products from silently collapsing. This section explains why the very discipline that makes synthetic biology powerful is the same machine that historically hollowed out the engineer's own creativity. Elphick's chapter argues that this hollowing is a design choice. Nothing in the biology forces it. The field built the arrangement, and the field can rebuild it. 3 partitions do the work: standardization, abstraction, and the designer, engineer, technician hierarchy. Each partition buys the efficient delivery of known ambitions, the drop-in fuels and drugs, by subcontracting invention away. And here is the tension that carries the whole argument. The very partition that carries the whole argument, the very partition that scales what you already anticipated, is the same partition that forecloses what you did not. So the primitive is this: creativity is slack, that you deliberately architect into the structure. you build room for it. Tilt the hierarchy, overlap the skills, molt the standards, fail confidently. Otherwise, the transformative futures that the whole enterprise promises simply never arrive. This section explains why control over life is a category error, one that only shows up at the scale of a human lifetime and dissolves the moment you widen the frame. The chapter uses time as an instrument of humility. The cyanobacteria that synthetic biology now rationally designs for biofuel are the same organisms that oxygenated the atmosphere and laid down Earth's crust. Their reach spans picosecond electron transfers at one end and millennia, long stromatolites at the other. Katz and Iwasaki answer engineering's control hubris through engineering futility rather than through critique. They deliberately break efficiency so that life messes up the linear logic of design. In the arc of the book, this is the pivot. It turns from what synthetic biology builds toward whose clock the very act of building registers on. Zoom out far enough, and the whole Anthropocene is one thin layer of rock. This section explains how the 11th chapter attacks synthetic biology's founding premise, using time as its only weapon. Synthetic biology assumes life is something to be rationally designed, standardized, made efficient. Against that premise, the chapter aims a single weapon, timescale. It picks cyanobacteria, Earth's oldest and slowest engineers, the ones who oxygenated the atmosphere and laid down its rock. Then, it deliberately engineers futility into them, instead of utility. Into them instead of utility, and the field's whole logic of control inverts. The generative move belongs to DeLanda. An observer whose clock is vast enough sees flows rather than organisms to command. In DeLanda's words, he would worship flows, unlike us, who worship rocks. Control is an artifact of the human time scale. Rescale the observer, and hubris dissolves into humility. The chapter's place in the book is the artist's counterframe to the engineer's. Here, it is time that does the destabilizing rather than argument. This part explains how critique can work by rescaling a variable instead of answering a claim. Critique need not answer a claim. Rescale the variable the claim silently holds fixed, and its buried assumptions surface as choices. Synthetic biology fixes one temporal window, the near future, quick wins within a decade. Iwasaki and Katz's cyanobacteria art dilates that window. It stretches from millisecond metabolism to the geological millennia in which the same organism laid down Earth's oxygen and living rock. Under that sweep, the field's promise of useful products stops being background and becomes a contestable choice. The engineered microbe made to secrete fool's gold turns utility itself into the question. Here is the primitive. Move the scale parameter and the invisible frame becomes the foreground object. This is what art does that argument cannot. This part explains how Chapter 12 of the book turns time into a critical instrument for judging synthetic biology. Widen the one variable a field holds fixed, and its unspoken assumptions surface as parochial. Synthetic biology silently sets its clock to the decade, to quick wins, to near-future products. Calvert's move in Chapter 12 is to sweep that parameter. At geological time, DeLanda's flows dissolve every utility claim into insignificance. At the metabolic scale, Oron and Hidio's cyanobacteria digesting chips into fool's gold build something deliberately useless, indicting the assumption that biology must be made useful at all. Time becomes the critical instrument. Consider the chapter's role in the book. Art performs its critique by varying the frame rather than by argument. The artist reaches the one verdict the critical designer cannot reach. The designer has already assumed a future user, and so the designer cannot say what the artist can say, that the technology may simply fail. This section explains why a strand of synthetic biology and a phrase of music are, at the deepest level, the same thing seen through different senses. Structure is channel agnostic. A plasmid and a phrase of music are the same object routed through different senses. Sonification's real claim is that the flat visual, GATC string, discards a hierarchy. The ear can climb nucleotide, part, function, whole. Each of these is a level. The composer navigates by changing playback speed, so rate becomes zoom. That is the gear shift. Look next at the operation itself. The chapter's place in the book, the artist's cut and paste on sound, and the biologist's cut and paste on genetic parts are the same operation. That sameness is why the collaboration yields knowledge rather than decoration. The surprise that carries the whole chapter is this. A representation is a discretionary choice of channel. Pick another channel and you recruit perceptual faculties. The default one wastes. This section explains the core generative move behind turning biological structure into sound. The authors state it plainly, the representation of structure as a temporal flux. A plasmid structure is static. Hearing parses only what unfolds in time, so the collaboration manufactures a time axis the data lacks. It does this through two levers. The first is playback speed, the gear shift that sweeps from nucleotide to part to function across zoom levels. The second is decay, where a sequencing chromatogram's fading trace maps onto a room's doom levels. The wager is that structure is substrate neutral. The hierarchy that folds a protein and the one that builds a musical phrase are one abstract object. Routing that object through the auditory channel, which evolved to read structure and time, makes latent biological structure perceptible. The right framing here is a change of representational basis. Sonification is not a recoding of letters as notes. It shifts the basis in which the same structure is represented. This section explains what abstraction means in Shifter's account and why every stand-in leaves something behind. Abstraction is representation, a stand-in swapped for the thing. DNA becomes a part, a cell becomes a chassis. It does not stand opposite to the real. Shifter's single move collapses the two words into one. To abstract is to substitute an engineering idea for living matter. Every substitution is lossy. Something of the original always stays behind. The chapter installs this ledger so the book can audit synthetic biology's founding ideal by one question. What does the stand-in withhold? Chafe and Leguia's sonification is the mirror representation deployed to question representation. The contrast that fixes the structure is this. A stop sign represents by command, a painting by likeness, a part by analogy. Same act, different loss. This section explains why abstraction always costs something. Abstraction is representation. And representation is a lossy standing in. A stand-in earns its use precisely by discarding some of the original. This is the single primitive threading. The chapter in Shifter's account, DNA becomes a part, a cell becomes a chassis. Not to describe them, but to make engineering tractable. Every such trade buys a capacity with a subtraction. The loss is constitutive, not a defect. What synthetic biology's mechanical metaphors subtract is the specificity that makes living things alive. Chris and Mariana's sonification is the mirror move, representation turned on representation. It lets the listener choose what to hear, and so exposes that each choice both conveys and withholds. From this, you can predict any abstraction's hidden cost. Ask what it withdrew from. This section explains why synthetic biology inherits modernism's failure mode. Le Corbusier called the house a machine, a habitat, a machine for living. He treated the dwelling as rationalized apparatus and its inhabitants as standardized parts under the architect's top-down control, eschewing diversity, context, and self-organization. Ginsberg's move is a chiasmus. Invert the phrase and you get the living machine. Venters Cynthia, life engineered from the top down. Same ideology. New substrate. The claim the rest depends on is that synthetic biology reprises modernism's design grammar and therefore inherits its failure mode. The international style promised a universal fix, and once built worldwide, it produced the opposite. To read a designed organism as a machine for living rewritten is to predict, structurally, that context-blind top-down control will misfire the same way again. This part explains why the strictly non-living machine is only a parenthesis in machine history. Read machine history as 3 regimes. First, ox and plow, living engines. Then fossil fuel mimics copying their motion. Then engineered life returning as a third form. Against that arc, the strictly dead machine shrinks to a 2-century interlude, a brief exception rather than the baseline. The inversion that carries the whole argument is this. A machine defined by its maker's intent is one regime. A living machine smuggles in its own telos, survival, so you build it once, then share authorship with Darwin. It replicates, adapts, and evolves out from under its designer, coupled to an environment it reshapes. Where the book's earlier chapters ask whether life is like a machine, this one asks what breaks when it is one. This section explains why a living machine is a contradiction by naming the single axis that evolution and design sit at opposite ends of. Start with 2 animals. A mule does its job for 1 generation and cannot reproduce. A horse reproduces, evolves, drifts. Calvert's chapter turns on exactly this difference. To call something alive is to demand that it evolve. Yet to engineer something is to demand that it stay put, predictable, controlled, yours. So the living machine is a contradiction in its own terms. Evolution and design are 2 poles of one axis, generativity against control. You buy novelty by ceding control, which is directed evolution, the best designer we have. Or you buy reliability by killing reproduction, which is a mule, a disposable synthetic Y chromosome. That is the chapter's role in the book. It names the trade that every synthetic biology approach silently takes a side of. This section explains why a mule holds an entire argument about living machines. A mule is that whole argument in one animal. It is bred for its useful traits, stronger and hardier than either parent, and sterile. A disposable biological system, good for exactly 1 generation. Calvert's chapter turns on the tension the mule exposes. Life is defined by evolvability. NASA's working definition calls it a self-sustaining chemical system capable of Darwinian evolution. But design demands control, and evolution buys its open-ended novelty precisely by discarding control. So a living machine is merely a contradiction. Let it reproduce, and it drifts past your intent. Forbid reproduction, and you get a mule, controllable, predictable, no longer harnessing what made biology worth engineering in the first place. Design and evolution are the two poles of one axis, the axis running from control to openness. This part explains a single molecule whose meaning flips entirely with its label. Isovaleric acid reads as delicious in Swiss cheese and repulsive as sweat. Identical chemistry, opposite valence, and the valence is fixed entirely by the label you are handed. That is the whole chapter. The line between body and food, between disgust and delicacy, between self and microbe, is drawn by context, not by substance. Agapakis and Tolias make this material. They culture cheese from swabs of their own skin, armpit, and toe and nose. The body is already a symbiotic colony, one that outnumbers its own cells 10-1. And cheese is that interior turned out, made legible to the nose. The book's move is against the sterile, isolated vat. It puts symbiosis itself forward as the engineering paradigm. This section explains why cheese and body odor are the same thing, and what that identity reveals about the boundaries synthetic biology draws. Cheese and body odor are the same molecule, isovaleric acid, produced by Brevibacterium, whether it lives between your toes or on a Limburger rind. The chapter's move makes the point undeniable: culture cheese from human skin swabs, and the identity can no longer be denied. The chemistry is fixed and identical. What separates the delicious from the disgusting is the label. Told a vapor is Parmesan, the nose accepts it. Told the same vapor is vomit, the nose recoils. The molecule never changed. The body reads as an ecosystem, its own cells outnumbered by microbes roughly 10-1. The bounded self is a fiction, and the phrase inside out names that reveal. Here is the generative pattern. The boundaries synthetic biology polices, clean against dirty, self against other, art against science, are cultural cuts laid over continuous life. They are drawn by us, not found as seams in nature itself. This section explains why disgust is the lever this chapter pulls. Disgust tracks classification. It fires when something crosses a category line, whether or not that thing can actually harm you. The same skin bacteria that are clean on you become dirt when swabbed into cheese. Nothing about the bacteria changed. They only crossed a category line. This is Mary Douglas's idea of matter out of place. A shoe is fine on the floor and foul on the table, the same shoe either way. The foulness sits in the location, not the object. Ginsberg's chapter runs one artifact as the probe that makes the rule fire. The human body cheese of Agapakis and Tolias, cheese cultured from bacteria, swabbed off human skin. The move the whole book turns on is this: synthetic biology's clean circuit diagram. Abstraction hides that we are the porous bacterial biology it means to engineer. Provoke the disgust reflex and the boundary it defends. Self against microbe, clean against alive, shows itself as constructed and therefore as redesignable. This section explains why disgust tracks a broken category rather than a harmful substance. Disgust is a measurement of a violated classification. The object it recoils from carries no poison. What has gone wrong is the category, not the thing itself. Ginsberg's anchor is the body cheese made by Agapakis and Tolias, cultured from swabs of armpit, nose, and toe. Its bacteria are no different from the bacteria in any cheese. Only their crossing of the skin's boundary manufactures the horror. This is Mary Douglas's idea of matter out of place. Dirt is not intrinsic filth. It is a symbol lodged where the classification forbids it. The chapter is the book's lever against synthetic biology's dream of uniform, decontextualized parts. The body is porous, symbiotic, 3 pounds of foreign life. An engineering vision that eliminates diversity founders on it. Here is the generative flip. We are no longer what we eat. We eat what we are. This section explains why the epilogue refuses to conclude, because its object refuses to hold still. Biology evolves. It resists the fixity that design presupposes. The future does not yet exist, so it resists closure. The faithful authorial move mirrors the object, withhold the synthesis, keep the space open, provoke rather than settle. 3 threads carry the epilogue. Biology's willfulness defeats the engineering metaphor. Design is inescapably value-laden, asking what could be rather than what is. And the future is inhabited rather than predicted. The failure mode, named throughout, is the drop-in replacement. Synthetic biology as a disruptive technology that also promises to disrupt nothing, a phrase from Chapter 3, slotting bacteria into unchanged habits. Anti-closure is the thesis, the form matched to a mutable subject. This section explains the book's epilogue and why it refuses to conclude. The epilogue refuses to conclude, and that refusal is the thesis. The book's diversity of voices is deliberately not collapsed into one verdict. Non-closure is the product itself, the chosen deliverable, and reads as a failure to synthesize only from the outside. 3 threads carry it. First, biology evolves, so to design it is to design with Darwin in Chapter 15. Second, engineering's stable substrate dissolves, and design becomes inescapably value-laden, moving from the question, what is, to the question, what could be. Third, synthetic biology, barely existent, yet, is inhabited as future and fiction. The generative move underneath all 3 is the same. Withhold closure, provoke the next question. The success metric is a reaction in the reader. The sentence, I need to go and think about that. What transfers is the impulse to keep thinking, not a settled answer. Here, frugality is refusal rather than compression.